I'm Amanda Egan, the Director of Patient Engagement and Clinical Programs for the RUNX1 Research Program. Really glad that you're all here. Today we are going through a couple hours of different topics with a focus on a journey towards a cure, creating the RUNX1 research plan together. We have multiple presenters that I'll introduce as we go, but we're really thrilled that we have a couple of patients, a scientist, and then several RUNX1 team members with us today presenting different parts of the session. So today I'll start by talking through the goals of today's session. First, we will review community input on target priorities for RRP. We'll review patient-centered outcomes research principles. Many of you have been a part of these discussions, so we will quickly go through those. And then we really want to talk about proposed approaches for how to do this well with RUNX1 FPD. We'll review the basics of RUNX1 FPD biology and the research targets that RRP is thinking about specifically. And finally, we'll talk about RUNX1 research programs, research strategy across the next several years. So let's get started on the community input on priorities. Many of you may know that I've had the pleasure and fortune to talk to many patients one-on-one over time and in small groups. And we've held a series of workshops that really led up to this final culmination of hearing from patients, hearing from scientists, clinicians on, what questions do they have for in terms of RUNX1 FPD, research-wise, education-wise, how to manage their own health in the clinic. But I'm really going to summarize really quickly what we heard as themes on these priorities. So one theme is that really there's a need to increase the knowledge of RUNX1 FPD biology. So this is, research, understanding of the biology of the disease. It's spreading the word and sharing what we do know on this on RUNX1 FPD in terms of the biology and understanding of how it leads to or can lead to cancer in patients. And so that's important for patients as well as clinicians and scientists across the community. Treatment development, obviously, as many of you know, we are really focused here at the RRP on developing treatments and specifically interventions to intercept cancer before it intercept cancer before it starts. And so we're glad to hear from you all that that's really at the forefront of your minds. Clinical care recommendations. As you may know, unfortunately, there aren't standard consensus guidelines that are out yet. And I believe we'll talk a little bit about this, but RRP is really focused on this. And we've just recently funded a young early career investigator to help pull this together, at least here in North America. And then we'll keep working more broadly. And then home health and well-being management recommendations. And this is a big topic that covers a lot of things that patients seem to be thinking about, as we've heard from you around, how do I manage my family? What do I watch for? What can I, how best can I sort of parent my children to be sure they're safe? All of that sort of stuff. And we really hope that through these clinical care consensus guidelines, those we plan for those to be part of that. So I'm glad to know that we're on the right track there in terms of what we're hearing from patients. And then finally, are we okay there? Yeah. And then finally, social and emotional well-being support. And as you all certainly know better than me, this RUNX1 FPD comes with a lot of stress and guilt and worry. And we definitely hear you and recognize that that's a priority for our patient community. So just to quickly group these, we see the first two as really research and education focused. The next couple really focused on a need for these consensus guidelines and education. And then that third one, we're thinking about new programs and new outreach programs. And so these are topics and focal areas that we'll really be thinking about and planning for across the next several years. Today, we're really focusing on discussing this first group of things, that sort of research areas and research priorities. So that's where we'll go today. So Now I'm going to get into, and by the way, I forgot to mention, feel free to interrupt me at any point that you have questions or want to touch on any of this further. We don't want to leave you hanging, so feel free to let us know. So really quickly, a lot of you have heard about this throughout the last several years, but patient-centered outcomes research is sort of not your traditional research. It's what you're seeing in this little diagram here is the traditional research process where, you begin from identifying and prioritizing research questions, designing studies, developing the proposal, collecting data on a study, analyzing that data, on and on. But what patient-centered outcomes research is, what it is slightly different. Instead of bringing patients in really at this data collection phase where patients come in the clinic, have their blood drawn, answer questions, patient-centered outcomes research really involves patients at every step of the way, from identifying and prioritizing those questions that patients have to, sitting in on design and providing perspective to ensure that research is designed in a way that's really feasible for participants to participate in. All the way to hearing about what data is being collected and what's being found and sort of providing input on what patients might think is going on in terms of that, the results of the data collection. So that's how PCOR is different than traditional research in one way, in terms of breadth across the sort of research life cycle. But in terms of depth, it can vary, right? So in terms of if, going back to this image, certainly patients can play a role in interpreting data, but in terms of like analyzing data, we wouldn't necessarily need patients to lead that piece, but perhaps they can participate in discussions and learn more. So this diagram really represents the sort of levels of depth patients could play, and actually scientists can play. Everyone can play in the PCOR research setup. So in terms of from the lower end of learning and participating to all the way to collaborating and leading. So we see PCOR as not a one size fits all. So what we're going to get into next is really talking about, well, okay, so what does this mean for RUNX1 FPD? How can patients really get involved in these different phases of research? And if you were at Princeton in, when was that, October? This past October, we actually broke out into working groups with scientists and patients really discussing where along this research trajectory can patients realistically partner with scientists. And so we've taken your input and we've really sort of laid it out across different activities that the RUNX1 research program conducts for our planning across the next several years. And so that's the next part that we'll sort of get into. And what I'd love for you all to do is really listen and think about, depending on what role you play in this world, really think about how feasible that is for you. Is it feasible for me as a patient to be a part of these design discussions? Is it feasible for me to perhaps disseminate what I learned to my peers and my family members? Because you'll hear us talking about that, but we really want to know is that, can we do this? Is this going to work for everyone? So that's what our small group discussions will be about later. So let's see, where am I? I'm now jumping around. Any questions so far? No. So what we'll go through is a little bit about the different tools that we're setting up to really help everyone in this process. The grant making process. So as you might know, RRP partners really closely with other funders like the Leukemia and Lymphoma Society, the Mark Foundation for Cancer Research, and we we offer grants to scientists. And so we'll talk a lot about how we're thinking about involving patients in that process. And then finally, we'll get into areas of research that we at RRP have really gotten into and how patients and scientists can partner together there. Okay, so let's get started with the tools. I am going to be quiet. This part will be presented by Mrs. Georgie Blackburn. I'm looking for your face, Georgie. There you are. Okay.
Thanks, Amanda, and hello, everyone. Involving patients more deeply in research development is an exciting concept that began with the PCORI Award a few years ago. As we move forward with this initiative, we plan to enhance the existing tools and resources to support patients and scientists. We need to be ready to partner on research so solutions may be found to the RUNX1 puzzle. Our toolbox, if you wish, includes 5 areas to reach our goals. Number 1 is training. A set of training videos were developed to help patients, family members, clinicians, and scientists to better work together on research development and implementation. These videos help patients and all interested parties to understand the biology of RUNX1 FPD and the fundamentals of research, and how to apply patient engagement approaches to research development. We plan to streamline these videos that are already online and share widely among all communities. If you haven't viewed them to date on the RRP website, please do so. Number 2 is the glossary. We will update the glossary and frequently asked questions on RRP's website for broader consumption. Our website houses a glossary of technical terms and a set of FAQs about RUNX1 FPD and the disorder itself. We as patients all understand what's happening in our particular bodies, but as a collective, there's much more to it. Many patients, including myself, have requested the information on a level for broad understanding of technical terms. Very, very simply, we are not all scientists. However, we need to know what's going on in our body. So this is going to be a major objective. We will refine the information so that patients and family can better understand the disease. And in that respect, we welcome and request volunteers to help provide feedback, identify what is being made to be clear and what is still missing. Please let the RRP know if you would like to help, and I hope many of you will. Number 3, answering patient questions. In the last couple of years, we patients were asked to submit our questions about RUNX1 FPD. Dr. Lucio Castilla, our next presenter, shared a number of these questions at the scientific conference last October in Princeton that Amanda alluded to. We also worked in small groups at that conference to discuss what answers to the questions are already known and what there is still to be learned. For those of us attending, it was an enlightening experience to have those discussions. Our plan is to share what is known with the community in communications, so we are all on the same page with disease understanding and the research progress to date. Number 4, translating science. Earlier this year, the RRP emailed out 2 important communications, translating studies completed by Dr. Cindy Dunbar and Dr. Morty Ponce's lab. Knowledge is power. The RRP will continue to create and share these updates on recent research findings and the outcomes learned moving forward. Lastly, annual meetings. Collaboration is also key. We'll continue to have annual meetings such as this, as well as scientific meeting takeaways to keep all parties informed and aware of what is planned in the coming year. This will be a busy, empowering year. Next, Dr. Lucio Castillo will discuss patient engagement with RRP's research grant-making program.
Thank you, Georgie. And as a foundation, the RUNX1 research program offers significant research funding to scientists that are studying RUNX1 FPD, with the goal of helping to progress towards cancer prevention, as well as other quality of life considerations for the RUNX1 FPD patients. So one way the RRP influences and promotes RUNX1 focused research is through the requirements of that are associated with these awards. So let me show you the process of grant making. First, the RRP would put out a request for applications, or also called RFA. This is a document that explains the sort of research they would like to fund and what the scientists need to include in an application. They also include requirements for the period of time that the project runs, and a set of numbers of reports or meetings with leaders in the foundation, like with Dr. Katrin Eriksson, for example, to discuss the process and the progress during the grant period. So next, the RRP shares these applications with a review committee. This review committee is composed by leaders and scientists who understand very well the ranks 1 biology and ranks 1 FPD, and it's a group of scientific advisors of the RRP. Together, they review the study goals, design, and plans for data analysis, and they rate applications on a number of preset measures. Finally, they come to a grade or a score for each applications, and they rank them. And the RRP and any funding partners can select the highest review applications for funding. Then, of course, the grant, once it has been awarded, there is a period of time in which the research activity occurs, and they are in communication with the foundation at this time. And finally, there is a grant administration aspect to consider. These would include reports as well as payment of funds at certain scheduled times as the milestones promised in the applications are met. Within each one of these phases, we see and learn in the Princeton meeting in October from others, different ways to encourage the involvement of patients in this process. So for example, during the RFA announcement, and the RRP could delineate specific patient-generated research questions and patient-identified concerns as priority topic areas for applications. So also the RRP could require scientists to include a late summary of study proposed in their applications to share with the patients to include who are included in the review process. This is a common use in other funding in agencies and other formats. So this would be something very simple that could be implemented. So during the proposal review and selection, 1 or 2 patients could be assigned as part of the review committee for each grant, and they could participate in the evaluation and the grading for each grant. So when in the next step, when the research activities take place, the researchers could have 1 or 2 meetings a year with scientists and patients advisors to discuss the research design and gather patient input on recruitment, consent forms, data collection plans, etc., as it applies to each grant. So in addition to the specific meetings with the RRP and with the patient advisors, also they could have meetings with the open group of patients where they can summarize the research progress and the significance in either of the basic research project or the translation to the clinic, how this impact the FPD and ML. And as appropriate, this meeting may also include also a discussion of the initial results and the interpretation and planning for sharing finding data with a wide group of patients and their clinicians also. So finally, in the grant administration aspect, the RRP could require that periodic reports include updates written for a patient audience to track the progress and the comments that are made by the RRP. And finally, the RRP could require presentation at patient meetings or a webinar, for example, at least once during the funding period. So these are ideas that we have about how we can incorporate patients in these activities. And we look forward to hearing during the small group discussion later what you all think about these approaches, these ideas, and how feasible they are for everyone involved. So next, you will hear from Sean about patient involvement in the RRP-led research.
So Lucio was introducing us to the process for funding and grant funding to people outside of the RRP, but there is RRP led research that's also been going on. One of the examples of that has been the development of the RUNX1 Patient Data Hub. I'm assuming if you're on the patient side, you've probably had some experience with this already. It is up and running in its initial form. There were a number of us, including me, that were part of the advisory bodies when we were looking at different aspects of the data hub. And as the data hub expands, there will continue to be opportunities to be involved in that information. But let's look a little bit at the data hub and the different elements that went into creating it. First part, and something that we need to look at, is our recruitment refinement. We have recruited sort of the people that we know into the system, people that have already reached out to the RRP, and we'd be looking for ways to kind of expand that recruitment. You know, is it other family members? Are there ways to reach people that may be part of the community but don't know it yet? We need within the system, for it to be successful, we need a certain number of people for significance in those statistics. We also need to identify new research topics. That will be something that is prioritized based on the interest of patients that are part of the study. And we also have to look at building that up gradually. We don't want to wind up with a little bit of fatigue in terms of both the prioritization, identification, and then the different surveys that might be associated with the hub. The surveys is another element of it. We have existing surveys that we worked with and adapted to create the hub to begin with. But those surveys can be expanded on, and as more specific questions come up or based on prioritization. There is an electronic consent form. And if you are a RUNX1 patient, you probably have signed more than one consent form over the time that you've been aware of your diagnosis. But with the basic one that we have, again, as things change, as there are additional questions that may be asked, we might work with patients to try to figure out language for the new consent that we'd be asking for. Data accumulate for sufficient numbers for statistical analysis. The results of that will be something that will be coming out, will need to be interpreted, need to be understood. And as we have found out from even from the NIH studies that are going on, the more information we have, the more we realize there's other things we'd like to look at. And finally, that once we have those results, how do we share them? We can share them within our own community, then also look at the ways to expand beyond that community to find new opportunities to people that might value the information that's been found. So all of this is in line of getting to research to get to treatment development. And that's the next section that I'll talk about briefly. There are a lot of scientific teams that are looking at clinical trials. A lot of that information is underway, may not be available yet. But even as those studies come online and new studies are formed, it can be a really valuable thing to have patients involved in kind of looking at what those studies are. And this is basically an outline of how those studies might come together. So for example, in the early phases, we have the basic research and early discovery. That's the time frame when we are looking at what are the things that might be of value? What are the things that are of interest to the scientific community that have resonance with the patient community as well. There are a lot of questions that come up when you're creating a study and trying to understand what the benefits might be. We actually did a sort of test run of some ideas about some treatment studies involving medications and how to identify what would be a value there. And it was really interesting through the course of that to discover that the scientific community had focused on the set of medications that might have the best chance at results based on their previous experience. And the patient community was rather focused on how many times do I have to take this? Do I have to go see the doctor for it? And the quality of life issues that might be associated with that. So, part of choosing that treatment is really trying to balance out both the potential efficacy of a treatment, but also the potential use and benefits that might be recognized by the patient community as well. Once we get into clinical trial development, those questions become even more significant because you have to make it real. Then, are patients traveling for this? Is it something that can be done remotely? Can they, get blood drawn from a local agency and have it sent to a researcher? Do they have to go to the research facility, as with the annual NIH studies? So how does all of that work? How are the patients that are going to be involved in this going to be recruited? It's similar to the RUNX1 patient hub. What kinds of ways can we find it to make it that clinical trial, make people aware of it so they can go participate. And then also, again, as with the hub, the consent form language, they are very confusing. What are the things that we can do to make them a little bit more patient sensitive, so that people really understand what they're signing up for. Then once the testing has been going on, there is data review. The Food and Drug Administration is going to be looking at the data that is collected as well as the initial clinical study. But in the final round, looking at that, the results, and apparently now, according to the notes Amanda gave me, the FDA does require patient input, which is a really great thing. Not something that I've experienced in my previous years of studies, so that they're looking for that patient involvement as a really great thing. And then finally, once there is a treatment that's available, they do want to continue to connect on treatments and look at it after it's been used, see if there's anything that needs to change in the study, or see if there are new warnings or benefits that hadn't been initially anticipated that they would want to realize. So all of that is it's a pretty long process, going from the early patient involvement to defining helping to define the kind of setup for research projects all the way through to actually using the products. I think we're going to go into some small group discussions now in a breakout session.
Great. Thanks, Sean. I did want to mention, I realized while you were speaking that this is very US centric. There are obviously regulatory, this image that we made for you, Sean, is, we have FDA listed here, but obviously there are other regulatory agencies that operate very similarly in Europe and the UK and other areas. So anyway, just FYI, apologies, we didn't include those. But yes, thank you everyone for your presentations and your great engagement throughout all of these workshops and all of this work we've been doing, and looking forward to more of it. So as Sean said, what we're going to do next is we're going to go ahead and break out into small groups. Hopefully, I know Catherine has been working diligently, hopefully in the background, to make sure our groups are pretty even as we break out into them. We know that we always fully expect that not everyone can show for these events, and but we do have a great number actually, so that's nice. So what we're going to do is break out and discuss a few questions for about 30 minutes, and then we'll come back and share back a little bit into the large group. And so the questions that we'd like to discuss with you all are, what seems promising in this plan? Which aspects of involving patients in the design and all of these steps in research seem promising? Is there anything that seems infeasible in this plan that just wouldn't work for you from a patient perspective or from a scientific perspective, given what you know from that side? What are you concerned about? And, what are you excited about? And then is there anything that you'd add to this that's missing? Well, thanks everyone for your engagement. Can you all see the small group discussion slide?
Mhm.
So I hope the discussion went well. I think at this point, I'm going to just open it up for just a few more minutes for anyone to share from your groups a few points that sort of resonated or bubbled to the top. Just open it up to whoever wants to share.
We had a vote. This is Georgie. We had a very vocal group that shared a lot of concerns and a lot of good ideas. And Alex has taken wonderful notes, but a common denominator was frustration with physicians, having to educate physicians and how physicians react differently from one extreme to the other, being very proactive with platelets ready to do a simple filling in a tooth to not reacting at all when you have to do a reconstruction of an ankle. And so I think education, the more we can educate across the board to the clinical community at large, hematologists, will be helpful. And Alex has, I know, a lot of good notes that he can add to that.
Sure. Yeah. I mean, I think that was a big conversation point was, education to clinicians, even dentists. And then, the testing, what's appropriate testing. And I think another thing that I heard too, and I have notes on, which is, what seems promising about it in this plan is, really about the priorities of the patients. It was helpful to learn about the grant process. I think I heard also that the patient data hub process was fairly easy, and there's interest in knowing where the data is going to be used and how it's going to be used. So there's just some kind of overarching themes that we had during our discussion.
Thanks,
Thanks,
Alex.
Alex.
Sure.
And Georgie.
I can fill in a little bit just on that last note. We had comments that the data hub was kind of confusing to use, especially if you say that you're done, but then you want to go back in and edit anything. It's hard to, fix documents that were posted that were not the correct document. It was a difficult process because theoretically everything was completed. So finding a way to actually update an uploaded piece of information with the correct information was hard to do. On the promising side of things, the idea that that getting a grant also requires being able to present the information that your grant was based on to patients was really positive, as well as the involvement of patient in the clinical trials. I think on some of the infeasibility, there was some difficulty with the patient data hub, and sometimes the wording is a bit confusing. So sort of figuring out the process of going through on missing to add, again, in that positive side of ensuring that the discussions involve patients. When that happens, making sure that as patients, we do have enough information to help inform the clinical trials in a way. And then lots of, I guess, interest in seeing about the more opportunities for a clinical trial where there would be an in-person, but also a remote way. Telehealth telemedicine is a big thing now. So are there ways that we can tie into that and be able to participate in clinical trials without having as much of a time commitment and travel commitment to do it. Those are all some big points.
Amanda, I'll jump in about our group. We were really focused on discussing research and how patients can be involved in research. And there was consensus that we wanted to get involved in 3 main ways besides the data hub, of course. And one was big picture, like what generally are we choosing to invest in and why? And that could be something that is open to the group of everybody, all patients and caregivers, and perhaps something done anonymously as well, just to ensure that we get as many people to respond as possible. And then there was sort of the mid-level of people getting involved in an RFA, a request for application phase, where, okay, so let's say inflammation, taking a subtopic like that. And perhaps that would be something that would be more suited to a smaller group that we already have in existence, or a smaller group of patients that come together, so it's done privately. And then the third would be sort of this, not initial phase. This would be done by the experts as it has been done, but maybe like a second stage where if we were to have, let's say, a group of applicants for a particular grant, that at some stage we have this group or another smaller group of patients come together to look at a lay version of what those particular proposals are and weigh in with their perspective as well. So definitely there was interest in being involved in all different stages. And the only invisibility was, just that concern, which, what happens if this isn't necessarily representative of everyone's opinion? Because of course, when you only have certain people involved or certain people being more vocal, you run the risk of that. And how exactly we communicate this all and get this out there. Katrine, do you have anything else as our note taker to share?
No, I think you did a really nice job. I think that there was another, I had asked the question about, not wanting to overburden everyone with all this requests for feedback. But one of our group members said, well, it gives everyone an opportunity to weigh in, when they can and if they're interested. So we should, not limit our requests just because we don't want to burden people, because some people may have an interest in weighing in one area and another somewhere else. So I thought that was helpful feedback as well.
Mm-hmm.
Great. Well, thanks everyone for that great discussion and that input. And I'm glad we have the notes to take away and sort of think as we put this plan into place. We're actually now going to shift a bit, unless there are any other questions or comments related to PCOR and involving patients in this research. We're shifting to Dr. Katrine Erickson presenting about sort of the biology of RUNX1, how cancer develops, and these different points at which we're looking at intercepting cancer and with our research lines.
Great, thank you, Amanda. So let me attempt to share my screen here. Is that coming through? Okay.
Mm-hmm.
Okay, great. Well, I always think it's important to start with our goal. And this is really what we keep front and center every day. And it's a guide to every decision that we make. Okay, now you're seeing there's a weird wonky thing where the slide deck just wants to start moving. I seem to have a ghost. So sorry about that. And so, we were founded really to achieve this one goal, and that is to discover a cancer prevention for RUNX1 FPD. Obviously, we've talked about the importance of improving quality of life in RUNX1 FPD patients as well, but front and center is cancer prevention. Amanda asked that I review the basic biology of RUNX1 FPD and then our research strategy, which I will do, but I'm going to do it in a slightly different order. So I decided I'd start with our strategy at a high level, and then as I delve into that strategy, explain the biology along the way. So I hope that works out. And I'm happy to take any questions whenever they arise. So you can just interrupt me, or if you don't feel comfortable interrupting, just put it in the chat icon, and then maybe Amanda and Catherine can interrupt me. So I'd like this to be, a discussion. So hopefully at the end of this, I will have successfully conveyed, why, the why behind our research strategy, and simultaneously managed to convince you that it's really important that we hear everyone's voice, and that will help shape and inform our research strategy moving forward. So, but we have to all go into this eyes wide open, right? So we need to be aware that this goal is not for the faint of heart. Drug development is hard. There are a lot of failures, and that's mostly because biology is complex, especially human biology. And, here you can see I took from a recent publication in 2020, the average time it takes to pursue a new drug, and the average, the range in cost from anywhere from 314 million to 2.8 billion. And this is across many different therapeutic areas. So it's inclusive of oncology, cardiovascular, et cetera. It didn't actually have any rare disease studies. So I'm hoping that rare disease, if there was a study on rare disease, it would be cheaper because we have a smaller patient populations. And so the clinical study shouldn't be as expensive. But, and also I want to draw your attention to the fact that this beginning part of the, research continuum here at the preclinical stage and the IND stands for investigational new drug enabling studies, which are studies that are required by the regulatory agencies to prove in animal models that whatever drug you want to put into a human is safe. That cost is about 10% of the total. And that's a key point because what I'll show you in a moment is that that's where we need to play as a community. That's where we need to put our money. And then once we get something compelling, the rest will get taken care of. We don't have to spend that capital. Pharma, biotech, et cetera, can take that on. So that's really important. And the other really key point here is, as you move across these different phases, the risk or the failure rate begins to diminish. So you start to narrow and increase your understanding as you move from discovery to preclinical to IND and into the clinic. And so that's an important point too. And that brings me to this slide. So, and that's really a key part of our strategy, right? So as a community, our role is to de-risk potential therapies. We need to fund research that demonstrates we understand the biology. There's that ghost again. Sorry about that. Okay. We need to understand the biology of the disease and demonstrate that we do. And we also need to demonstrate that we know how to intervene. We have therapeutic approaches that make rational sense based on the biology that we understand. So that's really key. The other important point, and that's not depicted on this slide, is that we also need to show potential future partners that we are clinically ready. And what do I mean by that? That means that we understand the natural history of this disease across a large swath of the patient population, that we know who is in our community, where they are in the disease stage, where they are geographically. We need to know about symptoms, family history, even their preference of a therapy. So we talked about this already, right? What is this patient community willing to take on as a potential risk for whatever that potential benefit might be? How important is cancer prevention, and what kind of risk are you willing to take a potential drug? Those are really important questions, and we have to be able to communicate that to not only regulators like the FDA, because they really care about these questions, but also potential future partners. And so you can see here now on the slide that we are playing in that sort of early stage of the research process. That's where we need to raise money as a community, and we need to stimulate research and really de-risk a therapy. And then it gets picked up by pharma, biotech, venture capitalists. And then at that point, we can show we've got this full package. We know who the patient community is, we understand the disease, all of those things come into place and make us a strong partner. The other thing I wanted to mention is the importance of strategic funding, meaning we're thinking very thoughtfully about what research projects are going to address key questions that we need to fill. What are those gaps? So that, again, we can understand this disease, understand the mechanisms, and ultimately intervene. So that's really what I meant by strategic funding. The other is this, the importance of drug repurposing. You've probably heard about this. Drug repurposing is really valuable because you can reduce time and cost. What I mean by that is when you have a drug that has been approved and you can pick it up at your pharmacy, that particular drug has already been vetted by regulatory agencies. So you've already completed those IND enabling studies, even phase 1 studies where you're trying to figure out dose of a drug, that's already been done. So all of that work, all of that money, we don't have to worry about. And that's the beauty of drug repurposing. So RUNX1 or RRP has been really emphasizing the need or the desire to fund projects that are screening for drugs that are already available. So that's another way that we're thinking about reducing our cost and trying to move quickly and find something quickly. And then the other piece I'll say is, that we're always thinking about how do we multiply our dollars. And so we're always on the hunt for people who are like-minded, who believe in the value of the research that we're funding, and frankly, the high caliber of the scientists that we fund as well. And when they see that, we're able to successfully find partners. And so we've been really fortunate to have partners that sometimes will put in 75% of the total value of a grant project, and we'll only have to put 25%. So multiplying our dollars is a big deal and will get us to where we need to go faster. Let's see if there's anything else I wanted to say about this slide, but I think maybe I'll stop there. Any questions at this point? So we have, I don't know what is so goofy about these slides. So we do have a roadmap, and we're really paving the way towards this goal. At the top, you can see where I mentioned this clinical trial readiness that I was describing at the beginning. That's so important. We're thinking about that, and we're investing in projects there. We're very lucky as a community that we've got the NIH-sponsored natural history study. That is a ton of money. That study costs about $2+ million a year. So fortunately, we don't have to carry that on our backs. And so we're all very, very grateful for Paul and his team and the work that they're doing there, because that's just critical data. And again, that's a piece of that package that should we get to a new drug that we see has potential value, partners will say, oh, wow, we see all this data, we understand the disease, et cetera. The same goes for the RUNX1 patient data hub. So that's a different dimension of data where we're collecting data directly from patients, and that gets at some of those questions I talked about already, patient preference studies, really understanding what is this community willing to take, and what's that bar look like in terms of safety bar and quality of life, right? Are you willing to take a pill every day, or are you willing to take a shot every day? Maybe not. Maybe we have to kill those right off the bat, that kind of thing. The other thing I wanted to mention is that we're also, we've been investing in data aggregation from investigators worldwide. So we have a RUNX1 database. We've been collecting germline RUNX1 variants. In addition to that, that's going to expand, and we'll start collecting clinical retrospective data from institutions around the world. So we're making progress trying to collect as much data, aggregate as much data, so that we can move as quickly as possible. So that's that clinical trial readiness that I spoke about. And then there at the bottom of the slide, you can see the 4 different therapeutic approaches. And this is where I'm going to start to talk about the biology and what these all mean. But I do want to draw your attention to the fact that we've got 4 different potential approaches that I think are all very rational. And 2 of those, raising RUNX1 activity and decreasing inflammation, we're about to go into the clinic with 2 potential clinical trials in the next year or so. So it's really exciting. And those are studies that we definitely want to get patients to weigh in on and provide their feedback as they're being designed. And I'll speak to that in a moment. Okay. Okay. So Catherine has spent a lot of time creating these animations to help, and it's going really fast, shoot. So let me go back, maybe I'll do it manually. So let's see. So the metaphor here is that your bone marrow, and it's not stopping, that your bone marrow is meant to represent, the lawn, right? So the seeds are the hematopoietic stem cell, those green seeds, and those stem cells are responsible for making the green grass on your lawn. And those are essentially all the different blood types in your bone marrow that, you can make red blood cells, neutrophils, macrophages, etc. And so this is in an individual who doesn't have a RUNX1 mutation. Naturally, of course, that bone marrow, that hematopoietic stem cell in the bone marrow resides within the bone marrow microenvironment. So it's not just floating around in your bone, it actually sits in little niches, like little nests. And it's reacting and engaging with that environment. And that environment is shaping the stem cell, and the stem cell is shaping the environment. So there's crosstalk, just like soil is to a seed in the grass. Over time, in any normal individual, you can start to acquire mutations. It's a natural occurring event. Oftentimes, those mutations aren't actually beneficial to that grass seed, that stem cell, and it just dies and it gets replaced by a new healthy green seed. But sometimes, especially as we age, the chances of getting a mutation in a gene that might actually be important and be linked to cancer increases. And that's when you start to see these different color seeds here. And as we age, unfortunately, our bone marrow starts to get inflamed too, even in healthy individuals. And that inflammation is not really helping the green seeds survive. In many cases, that inflammation can help the weed seeds to survive. And now you're going to see this weed pop up, and that's supposed to represent, a clone, a hematopoietic stem cell that now has a new mutation, and it's forming the weeds, starting to start to crowd out, if you will, the green grass. And again, getting older and older, more inflammation, more weeds. And then, now, this is what we would call clonal evolution. So we're starting to see signs that this grass is not going down a good path, and you can acquire another mutation. So now you've got a really aggressive seed here that has 2 mutations, and that particular mutation can now end up driving this really nasty looking weed in just a moment. There it is. And now it's crowding out. Now we don't have much green grass left. Now you get what's called cytopenias, right? So now you don't have many platelets left, your red blood cells have dropped. Now there's signs that this green grass is full of weeds, and we're moving towards cancer, malignant transformation. And once you have all weeds, that's essentially blood cancer. And this can happen in sporadic, as we all know. People can get AML and MDS sporadically. And this is essentially what it looks like in the bone marrow. And interestingly, when you look at a patient with blood cancer, their bone marrow is incredibly inflamed. And inflammation seems to be a huge contributor to the aggressiveness of disease. So what happens in RUNX1 FPD? So RUNX1 FPD, we've got bluegrass and blue seeds. So a unique lawn. And in this setting, this is just, again, a blue seed represents a RUNX1 mutated hematopoietic stem cell. For many patients, about 50% of patients, go through life without ever having this bluegrass obtain or acquire any weeds. They do just fine. But sometimes we know the disease progression in many patients at the same time do start to get these bad seeds that turn into weeds and take over and crowd out the bone marrow. And so this is sort of meant to show the disease progression as we know it and understand it today. And you can see those nasty weeds coming up at the end. So what we know about RUNX1 FPD, of course, is that compared to the general population, there's a 30-fold increased risk of essentially having a lawn full of weeds, unfortunately. So of actually progressing to MDS or AML or even another blood cancer. So what can we do about it? So as I mentioned, we have those 4 rational therapeutic approaches. And the first one is to target the pre-malignant clones. So now I'm using, scientific terms, but essentially what that means is targeting these weed seeds directly. And so this would be, you essentially kill the orange seeds, the clones, before they become full-blown cancer. And that's the idea with this targeting pre-malignant clones. There are challenges to this approach. One is you have to identify a weed killer that can either kill all of those seeds that have different mutations, which you can imagine might be very hard, or you have to identify a weed killer for each type of mutation that pops up. So there are some challenges with this approach, but it doesn't mean it can't be done. It's not impossible. So this is one approach, and we've been investing quite a bit in projects that are looking to do this work. Oh, I see a chat. Someone asking a question.
Sorry, I was just letting people know that they could ask questions there if
Oh.
they want. Yeah. Sorry. So far, so good.
Okay. The other approach is to raise RUNX1 activity. So we know that with RUNX1 mutations, you're essentially, the stem cell has lower levels than normal of the RUNX1 protein. And because RUNX1 is a transcription factor and it controls a number of genes downstream of it, a number of proteins downstream, if you were able to identify anything that could increase or decrease, depending on what RUNX1 does, if you could have a drug that could alleviate that loss of RUNX1, that's another rational approach, right? Probably one of the most straightforward ways of going. So here, this is just essentially taking a blue seed and with a drug turning it into a green seed and having it over time take over, and now you've got a green lawn. So a fairly simple concept. There are some challenges with this approach as well as with every. It's not easy because this protein is such a complex protein and has so many downstream effects. But the good news is that we do have some really exciting developments there. Cell and gene therapy, another appropriate approach to consider for RUNX1 FPD. And this is really, scooping out those blue seeds and putting in green seeds or healthy hematopoietic stem cells to, again, repopulate the bone marrow with only green or corrected healthy stem cells. And then the final one is reducing inflammation. And reducing inflammation is really, just as it sounds, we talked about the soil and how important the soil is in cultivating the stem cells. If we could get rid of those stem cells, or I'm sorry, the inflammation, could we then create an environment where only the blue seeds want to grow and the weeds no longer find that soil comforting and an environment that they want to live in? So that's really reducing inflammation through drugs that would target the soil. So those are the 3 therapeutic approaches. I hope that visual helps you kind of think through that. So I'm going to start. Yeah.
Sorry, there's one question asked in the chat that says, in Lynch syndrome, one can take aspirin as a preventative, but it takes 10 years of this therapy to show any effect. Is this something being reviewed for RUNX1?
So I am aware of the aspirin data in Lynch syndrome, and we have not looked into aspirin yet. It has not been tested, more because it's sort of a broad anti-inflammatory, and aspirin is, yeah, I have not looked. I'll be honest, it has not been tested. It has not been tested. I'd have to better look at the mechanism of aspirin to see if it makes sense in terms of the mechanisms of inflammation that we see in RUNX1.
Isn't there an issue? I mean, I've always been told from when I was very young, not
And the
that I couldn't
platelets.
take
Yes.
aspirin because of the
Yes,
impact
yes.
on platelets. Yeah,
You're absolutely right, Sean.
yeah.
Yes, that's a big one, right? That's a big one for RUNX1 patients. You shouldn't be taking any blood thinners. Absolutely.
I have a question back to one of the slides, the four. I thought I'd heard from Dr. Liu that in terms of fixing cells and replacing them within the marrow, that the malignant or the RUNX1 cells seem to be outcompeting the improved cells. And I don't know if that's something that still holds some measure of hope, or if that's something that, I mean, it was a little, it was not great news to hear that when he mentioned it, but I don't know if there's other things going on there that might have different outcomes.
Yeah, no, it's a great point. And when I get to cell and gene therapy, I had a plan to mention Dr. Cindy Dunbar's work, and that really that is a big challenge we have to work through and figure out. And there are some ideas about how can we eradicate all of the mutated RUNX1 cells with using an approach like that, so that then you don't have to worry about that competition. But right now, we have to solve for that. We have to fund some research to try to find some selection markers, for example, and I can get into that. Yeah, it's an important point, and a disappointing finding, as you said. Okay, treating pre-malignant clones, the first one. So you can see here, we're still in the basic science and discovery mode, but let me share with you at least. Is this not going forward? Let's see, there we go. What we've learned. And I think for a lot of you, this will be pretty familiar. We know that those, the most common first hit, so those orange seeds that drive the weeds, are B core mutations. And that was thanks to the work of several groups. Actually, it was really collaborative work across the world that helped to inform that, helped us learn this information. So you can see the most common secondary mutations, which has been shown in our RUNX1 database, but also through the natural history study, are B core mutations, TET2 mutations, and DNMT3A. These are all names of genes that have been mutated, and those are the clones that we're most interested in when we think about trying to find that weed killer. Those would be the first targets that would make the most sense, since they affect most patients. And this is just a call to action, that if you're sitting on this call and you don't know whether you have any mutations in the bone marrow, it's really important for you to talk to your physician, or if you're part of the natural history study, talk to the NIH team and ask, do I have any secondary mutations? And can you tell me what they are? Because that'll make you that much more empowered to understand and follow this research, of course, too. And you should be monitored and surveilled. So this is, these are on the left-hand side are Dr. Anna Brown and Dr. Hamish Scott. They really led this collaborative worldwide effort to collect data from institutions in Europe, in North America, and Australia, et cetera. And they were the first to publish on the common co-occurring mutations in RUNX1. And then beautifully, when Paul's team from the NIH recently shared their findings on bioRxiv, they too saw the same thing. They saw B core mutations are the most common. It's always wonderful when two studies confirm each other. So, so we're grateful for the work that's been done in that department. So what does that do? That really informs research, because now we can go back and say, okay, anyone who's doing research for RUNX1 or for RRP, we need to make sure people are studying B core mutations and trying to understand what is it about these cells, and can we kill them directly with any drugs? So that's the idea here. A medicine could potentially just kill the weeds and leave the blue grass. So this would really be considered, this is what I would call a cancer interception, right? Where something comes up, you see a pre-malignant clone, and you're able to intervene and avoid cancer development. So it's funny, when I want it to go, it's not going. And so there you can see the weeds being killed. So here are some of the groups, and I just wanted to show you that we are committed to this therapeutic approach, and we funded quite a few groups to get at this. So one of the most recent studies we funded was a study being conducted by Dr. Len Zahn at Boston Children's and his partner at the National Institute of Allergy and Infectious Disease. And what they're trying to do is, instead of looking at one particular kind of mutation, is there a common change, metabolic change in those mutated, to go back to the weeds, in those weeds? Is there some common vulnerability that we can take advantage of and kill those weeds, whether it's BCOR, TET2, DNMT3A, or any other mutation? And so they're doing some really interesting work that spans not only using model zebrafish, but also partnering with Paul and getting some of the samples that you also kindly share via the NIH natural history study. So that's one of the most recent projects we funded, but we've also funded quite a few early career investigators that are also looking at different kinds of different common mutations and seeing whether through their model systems they can again identify drugs that could kill those clones. Now I'm going to go into raising RUNX1 activity. This is that idea again where with a drug we can change the bluegrass to green grass. And what's really exciting is that this we funded work by Dr. Alan Cantor initially in the preclinical discovery stage, which demonstrated that imatinib, a very old drug that has a clean safety profile, has been around for 30 years, can actually increase RUNX1 activity. And so Dr. Cantor and many colleagues, quite a large group of investigators, are working together to build this protocol. And my understanding when I last heard was that this will be hopefully IRB approved in the next few months, which means then they can start to enroll and run this trial. And that's being done through the NIH and in kind of collaboration with the natural history study folks, Paul's group. So very excited about that. We talked about the hypothesis, but there are other studies that we've also funded. One thing maybe that I haven't mentioned and I want to reiterate or make sure that I do is that I talked about how there's lots of failures in drug development. We certainly aren't taking the strategy that we're going to put all our eggs in one basket. So we really are trying to be thoughtful about making sure that we're diversifying our funding so that if something fails, we still have 3 more potential approaches that could work, as an example. And so there have been examples where things didn't work, and proteasome inhibitors you see there on the slide. Actually, on the next slide, you can see Dr. Horwitz, who's a wonderful SAB member now. He studied proteasome inhibition, thinking that maybe those drugs that are also well known could raise RUNX1 activity, and in his study that didn't work. So sometimes it's really nice to fail fast, so you kind of cross things off the list as well. So I mentioned Dr. Cantor's work. He's collaborating with Dr. Sung-Young Pai, who's at NCI, and the two of them are really leading what are called correlative studies with the imatinib clinical trial. And that is all of the lab research that's required to help us all that's not the right word, help demonstrate that the data or when patients receive the drug imatinib, that we have laboratory tests that actually show RUNX1 is increasing, whether it be through platelet count, platelet function. They're setting up platelet function assays, or even just good old Western blot, which is an assay that allows you to show whether a protein level is increasing. So they're doing all those tests so that we feel, oh yes, imatinib is doing what we think it's doing. And I won't go into some of the other examples here on this slide, but suffice it to say, we've got a great group of scientists that are thinking about drug repurposing and thinking about this approach of raising RUNX1 activity. More than thinking, doing, performing. Okay, this is decreasing inflammation. I think we've all heard about this, and we've felt that this is a really, I don't want to say it's a straightforward strategy, but it's certainly a strategy that makes a lot of sense, because there's a lot of clinical indicators that inflammation is a real deal in RUNX1 FPD. We know from bone marrow evaluations of patients that there are high levels of these pro-inflammatory proteins. And we know that these pro-inflammatory proteins have been indicated in other cancers, including blood cancers, as really bad actors and as promoting the aggressiveness of a cancer. We know that there is release of these inflammatory proteins from different cells within the bone marrow. We know from Dr. Speck, and I'll mention her work in a moment, that there are blood cell types that where if those cells are deficient in RUNX1, they are very reactive to any kind of stimuli, and they kick out a lot of pro-inflammatory cytokines. So there's a lot of compelling data that inflammation is really an area that we should be thinking about and focusing on to try to see if this approach would work. And we have been doing that. And I'm going to move forward. And a lot of the initial discovery around inflammation being playing a key role was done by work that we funded. Dr. Agarwal at OHSU, Dr. Speck. We did not fund Dr. Liu, but his work through the natural history study has also informed our understanding around inflammation. And I think you all have heard that there's these high levels of high prevalence of allergic conditions, autoimmune or autoinflammatory disorders, and all of that creates systemic inflammation and can affect that soil that I talked about. And so here you can see some of the papers. This is all work that RRP funded and that Dr. Speck has been able to contribute to the field and contribute to our understanding of mechanism of disease. And then even there's more investigators that we funded over the years who've contributed to our deeper understanding of this issue around RUNX1 inflammation. Interestingly, Dr. Reimacher's work, and I'll show his recent publication, showed that if you delete or reduce RUNX1 expression in the cells around the blood compartment, and you can do that with various tricks using mouse models, you actually don't promote a inflammatory environment or an environment that would promote this hematopoietic or this blood dysfunction. And so his paper, while it is from a murine model or a mouse, his paper really suggested that maybe most of the inflammation that's happening in RUNX1 patients is coming from the blood system, whether it be a T cell or a neutrophil or any other, or a macrophage, any other blood cell. It's from his study, it didn't look like it was coming necessarily from other cells in the bone marrow microenvironment. And that's a key insight and can help us as we think about therapeutic strategies. So this is an example of some of the pro-inflammatory proteins that exist in the bone marrow that have been identified. I will say in RUNX1 patients, there are a number of these pro-inflammatory proteins, which is making it complex to know which ones are the most critical, because there are drugs that can target actually each of these. The question is, if you target one, is that really going to affect the soil so much that it's beneficial? And that's a big question. But we have been studying and have been supporting investigators that are looking at several of these drugs, one being etanercept, and Dr. Lucio Castilla is online, and he's currently running a project to test whether blocking this pro-inflammatory protein called TNF could help get rid of some of these weeds or prevent these weeds from growing out. The same thing, I'm really excited about sirolimus. This is a drug that has been approved for many, many years. And it is one that can not only directly target the seeds and help the seeds reduce their inflammatory response to the environment, but it's a drug that can also reduce the inflammation in the soil at the same time. And interestingly, sirolimus is being used in the anti-aging world. And people think that this drug might have an ability at low doses to reduce that inflammation that I was showing you that happens as we age. And so there's a lot of work going into addressing that. And if you think a little bit about what's happening in RUNX1 patients and the bone marrow, there are some parallels with what you might call accelerated aging, right? That you're getting more mutations in the bone marrow quicker and at a younger age than the general population. And so maybe there is some really interesting overlap with that research at the same time. So in the interest of time, I won't review any of these at right now, but if you have questions, please. Oh, there are questions. Are there questions here, Amanda?
Well, while you're on this inflammation, the question is, are there studies on anti-inflammatory diet, sugar elimination to help prevent cell mutation? I think that's related.
Yeah, so the American Cancer Society has invested a lot of money in attempting to understand whether diet, exercise, and those things can reduce your risk of cancer holistically. And there's a lot of data to show that that is the case. So choosing a diet that reduces overall systemic inflammation, that you're keeping your sugars in check, you're not pre-diabetic or hopefully not diabetic, all of those are really beneficial for anyone, including, of course, RUNX1 patients. But I would probably say more important, if you have this higher risk of blood cancer or any kind of cancer, that is, you should really be thinking about reducing your overall inflammation through diet and most importantly, actually exercise.
The other question while we're here is ASXL1 related to RUNX1 FPD or mutations in ASXL1?
Yeah, there are some, if you look at Paul's recent paper, some patients had ASXL1, and I think that in our RUNX1 database that we also saw some ASXL1, but it's actually a lot lower than what you see in sporadic disease. So in sporadic AML and MDS, you see ASXL1 mutations are much more common. But in the RUNX1 FPD population, we're not seeing ASXL1 as one of those really common ones. Not to say that it doesn't happen. Anything else?
Nope, that's it.
Okay.
Thanks.
So anything I want to add here, these are, all of these investigators are conducting experiments and projects that are looking at inflammation from different angles. Inflammation is a massive area, and so there are lots of ways in which we can start to tease apart what's really driving malignant progression or cancer transformation. And so all of these fabulous investigators are conducting projects in that area. And then here is that pilot study I wanted to share with you. This is this concept around low dose sirolimus. We're just starting the development of a protocol. And Courtney DiNardo will be the clinical PI, and Dr. Agarwal, who really did the preclinical work to demonstrate that mTOR is high in the RUNX1 mutated stem cell population, and that when she treated RUNX1 stem cells in a dish with mTOR, she could actually rescue the platelet phenotype, so she could increase platelet count in a dish, and she could also change some of what's called myeloid skewing. So she could improve the balance in the different hematopoietic cells that are produced from the stem cells by treating with mTOR. And so her preclinical work really led to, moving forward with generating this pilot study. And so this is one of the studies I'd love to bring back to the community and get some thoughts around dosing and just design and recruitment, that kind of thing. And then I can't help but share that we were very fortunate to get grant funding from Chan Zuckerberg Initiative, and they have committed $2 million towards the goal of understanding, again, inflammation, but at the single cell resolution. And so we just actually had a call with Paul a couple of weeks ago about how we might all be able to collaborate so we could get patient samples and start to really interrogate, okay, what blood cells might be the bad actors and really driving this inflammation? How is the stem cell behaving, and what inflammatory proteins is it responding to? You can get so much out of the new technology that's now available, which is called single cell sequencing. And there's a lot that we can learn from those. There's a whole bunch of new technology that allows us to get into every single cell and understand what's going on within each cell. So I'm really excited about that. And so we, RRP, got some funding for that to help work with these researchers and make sure that the patient voice is part of this program. And I know we're down to the last bit, so this is where I'm going to have to race through. I think probably everyone's pretty comfortable with the idea of a stem cell transplant. And so my first few slides are really about replacing the green lawn with green seeds and green grass. And you can do that by an allogeneic stem cell transplant, where you take a donor and you use their stem cells and repopulate the bone marrow. The challenge with a transplant is it doesn't come, it is not a 100% safe procedure. And the first thing you have to do is you have to undergo what's called conditioning regimen or conditioning therapy. And that can be a combination of chemotherapy and radiation, where you wipe out all the blue grass, all the blue cells, so that the bone marrow is prepared for that new green grass that you're going to infuse into a patient when you do an allogeneic transplant. This process is scary because for a little bit, the patients are really high risk of severe infection, because you can imagine there's nothing left in the bone marrow until the green grass gets put back in and can grow. So that's one of the, so here you can see there's no lawn to protect against invaders like viruses and bacteria. And so now what we really, as a community, what we need to make sure, or what we'd like to see happen, is that there are gentler conditioning therapies that that directly target the seeds and don't require chemotherapy or radiation that puts a patient really at risk. So now the next step, of course, to a transplant is putting those green seeds back in without a germline RUNX1 mutation, and you get that green grass to grow. But the challenge there sometimes is you can get what's called graft versus host disease. And that's where those green seeds are now, because they're from a different human being, might start to attack your body. And those are some of the really critical issues that transplant patients face. You get skin rashes, liver damage, fatigue. And I know some of you on the call have had to go through a lot of this yourself. So there are ways to try to reduce that graft versus host disease through a close matched donor or sibling, but that can be hard when you're a RUNX1 patient to find an unaffected relative that would be a match. There are ways to deplete T cells to reduce that. So there are ways. I bring all this up, and I'm sorry, I'm going to skip through. I bring this up because I want to show why we're not investing here. So this is stem cell transplants are a therapy that's needed for many different indications. People with sporadic AML, MDS, even people without hematopoietic malignancies, people with blood disorders, need transplants. So there's a lot of investment from academia, from companies that are really thinking about ways to improve this process. And so I wanted to share with all of you that that's why we have taken the strategic approach. Let's keep our money for other RUNX1 specific issues and let everyone else spend the money there. And if you have thoughts about that strategy and you think else, or you think otherwise, please reach out and let us know. Okay, 2 minutes. I'm not totally done, but I'll go really quick. So that the last concept is this idea of correcting stem cells, right? And that's using CRISPR, or now there are some new gene editing tools like base editing, maybe you've heard of, or prime editing. And that's where you take those blue stem cells out of the bone marrow and you correct them using these gene editing tools, and you turn them into those green seeds and you put them back in. And it requires that same process of harvesting cells. So you have to do that conditioning therapy that I talked about, which is a challenge, and there's some risks, and we can talk about those risks in another setting perhaps. And then you transplant those cells back in. Here are some of the risks that I'm mentioning. Some of those blue seeds can be left behind. So then you might still have leukemic risk if we don't get rid of all those blue seeds. And then the other piece here is that this is getting to Sean's point at the very beginning, that we learned through Dr. Cindy Dunbar that if one, you can't correct, the technology today doesn't allow us to correct every single cell. It's just not that efficient. So what you end up is a mixed population of green and blue cells. You transplant those back in. Unfortunately, the blue cells over time start to crowd out the green cells, and now you're back to a bluegrass where you started from. And that's what we learned from Cindy Dunbar's work, and that was work that we funded because we had that question. Well, what happens if a few blue cells get transplanted back in? Are they going to take over? The hope was that the green cells would take care of them. They would take over and the blue cells would die. But unfortunately, that didn't happen. And so we have to figure out ways to solve for that, and that we have some ideas. And maybe in another setting, I can talk through that. And so here's Dr. Dunbar's work, which was really, really important work and really important knowledge for us to all gain. I'll end, if you'll allow me, with this other clinical trial that is in development. And this is a collaborative effort across 3 different institutions. Maybe you recognize some of these people. So Dr. Tim Olson is at CHOP. Dr. Matt Porteous is at Stanford. He's actually one of the big gene editing principal investigators out there. And then Dr. E.J. Spall at MD Anderson. And what they're doing is they're pulling together a protocol which will allow us to mobilize RUNX1 patients. Mobilize means give patients a drug. It's a drug. It's a growth factor, which kicks the stem cells out of the bone marrow into the blood. And then we can collect those stem cells, and we're going to do a few things with those stem cells. One, we're going to save 1 bag for potential autograft. What that means is if later in life that patient who had their stem cells collected starts to acquire mutations and it looks like there's weeds growing out, we can give them their healthier stem cells that hopefully didn't have any mutations. We can give those back and just repopulate a bluegrass, so to speak. The other option is to have 2 bags or more banked for potential gene editing. And that's where, if in the future the technology gets better and we've solved for some of these problems I mentioned, now we've got cells saved from that patient that are hopefully younger, healthier, and ready for that gene editing should it come to the point where we can do that safely and effectively. And then the final piece is that through that mobilization, we learn, how do patients mobilize? And that is really important because if we ever get to a point where we can do in vivo editing, probably a 10-year horizon, but in vivo editing means you don't have to take the cells out. You can just put gene editing tools into the bloodstream and they'll go and edit the stem cells in vivo. And it's sort of a one and done. In a perfect world, that would be what it would look like. So this protocol is going to address quite a few questions and will be important for any of those strategies in the future. And then I think I'll just end. Thank you all for staying with me. I won't go, this is our portfolio. Know that we're, you know, we're really thinking thoughtfully about all these different approaches and making sure we're diversified. And we do this with collaboration, disease focus, and data sharing always. Thank you.
Thanks, Katrine. Sorry we crunched you for time because we
No,
did.
I
was
feel
really
bad that we
good.
didn't get much Q&A. I'm sorry.
No. Okay. I for one can stick around a little longer. I know we're at the hour, we're over the hour. So if people need to hop off, we won't be insulted, but really appreciate this collaborative effort by Sean, Georgie, Lucio, Katrine, Monica. I know I'm not, who am I forgetting? Catherine, Alex, everyone really playing a great role to get this done. And I hope more of you, we continue to see more of you come. I'm really glad you all came to listen in. There are a couple of questions. Katrine, do you have a few minutes to stay on with for questions? And those who want to hop off, feel free. We understand. Okay, so I believe I saw the first one I saw was will there be research on protector cells going forward? I think that's protector mutations like is this TCL1A and that sort of thing?
Oh, yes. Very good point. So, and I think we may have thrown this in one of our newsletters that there was a publication by an investigator that was at our conference, Dr. Sid Jaiswal, who collaborates with Dr. Alex Bick. He was also in that publication. And there is a gene called TCL1A, and you can ask, if you're in the natural history study, you can ask to see if you've been genotyped for this particular gene. And what I mean by that is we all have variants in our genes, and these are called SNPs. So they're a little bit different than what we're talking about with germline mutations that cause a disease. SNPs are just sort of variations in the genome. And it was found by Dr. Jaiswal and Bick that if you have a certain variant, I think it's the T allele, you're protected from specific clones, specific weed seeds growing out. So let me see if I can say that a little bit better. It looks like if you have this one variant, there are certain weeds that won't actually be able to grow because you've got this protective variant. And I understand from Paul that there are patients in our natural history study that do have this protective variant. And I know that Dr. Jaiswal and Bick are working hard to understand whether there could be a drug that could give everyone the chance to have that protective variant, if that makes sense. So like a drug that would turn on that protective mechanism in everyone. So yeah, great question. And another thing to ask if you're in the natural history study, ask if you might have that protective variant. The one thing I will add about that is it doesn't protect against every mutation, but it protects against one of those really common ones, the TET2 mutation.
So this is a question I've gotten in the past. I don't think you can see it right now, but someone was asking about having this person's past stem cell transplant and their donor was 100% match. So this patient is fully engrafted. Does this mean they no longer have the RUNX1 mutation? And what does that mean for like sort of prevention and concerns going forward, depending on what your answer is?
Sorry, I got distracted for a moment. So this person had an allogeneic transplant and it fully engrafted, and they have, okay. Then, you know, it's hard. You can never say your risk is completely gone because we all have risk, but you certainly don't have the same level of risk as you did if your bone marrow was filled with stem cells that had the germline RUNX1 mutation. So that allogeneic donor provided you with stem cells that don't have that mutation. And so you were able to eliminate that RUNX1 FPD associated risk for blood cancer.
And what about the cells that remain? Are there any cells in the body that still have the RUNX1 mutation? That was another part of the question.
Yes. So basically every non-blood cell still has the germline RUNX1 mutation, but and there will hopefully be some more data about this in the future, but there is no evidence to suggest that those cells, certainly in the mouse models, that's what Dr. Reimacher showed, that having a germline RUNX1 mutated bone marrow cell that isn't a blood cell will somehow, taint the healthy blood cells you just got and turn them into cancer again. So that the understanding is that your risk is gone from that perspective. But yes, you still have germline mutations in your skin and your heart and everywhere else.
Thanks. I think I got all the questions that were written. Does anyone else, did I miss anything or any other questions from the audience? Yeah, okay.
Okay.
I saw a new message. Great info. Thank you. And then I also said, someone said that Trin, you do a great job of expressing your passion through cures. So nice comments. Thanks.
Yeah, thank you.
Okay.
Thanks, everyone. It's good to see you.
So stay tuned as we start to roll out our plan for engaging more of you in these discussions about design of research and research strategy. So thanks, everyone.